grow bacteria on artificial media. Later I will share a reprint, a journal with you all. It's a review of different kinds of new approaches that the researchers are taking in order to grow novel bacteria. Novel bacteria or rare bacteria. These are the ones that don't grow as common as the other bacteria on artificial media. So why do we want them? Why do we want to isolate? Because in the hope of finding something new, something with a biotechnological potential, something that can be useful to mankind. Okay, so that's why we have microbiologists that actually go around looking for novel bacteria. So they are also known as bioprospectors. Okay, so they will go around looking for novel bacteria, novel metabolites, and all that. But once you have them growing, then you have to find ways of preserving them or maintaining them. Of course, the easiest way is to keep transferring them onto fresh media after a few days. Okay, but sometimes you want to be able to preserve them so that you don't have to keep maintaining them over a long run. Okay, like for example, if you're going out for research, okay, you have isolated, let's just say, thousands of bacteria from the jungle. You cannot be maintaining all these 1,000 bacteria every 2-3 days. Okay, if you are doing that, then your whole job will just be maintaining the bacteria. Then you can't really do any research. Okay, so you have to preserve them. You have to find ways of preserving them. So there are short-term preservation and long-term preservation techniques. Okay, of course, usually when we isolate them, let's just say if we isolate them on plates, artificial media plates. So the best way is actually just to seal them. We will seal them with Pyrex tape, Pyrex film, and then place them in the fridge. Okay, so this one will probably last about 1 week. Okay, so why do we seal them? Okay, we seal them to prevent dehydration. We do not want the agar to dry up. Okay, so there are two factors that can cause the bacteria to die on the plates. First is the plate dried up, no water, and the bacteria dies. Second, the amount of nutrients available totally used up, then the bacteria also dies. Okay, so when you see colonies on the plate, doesn't mean they are still alive. Okay, they could have died. It's just that you are seeing what is their carcass, so two factors that will determine whether they will live long. One is make sure the plate is not dried up. And the second, make sure there are enough nutrients. So this is can also be part from experience. Okay, basically, if you know the bacteria is a copiotroph, then you know you shouldn't keep the bacteria too long, because even after 2-3 days, the nutrients could be used up. However, if the bacteria is an oligotroph, then you can safely say, oh, the bacteria will probably stay alive even if 1 week in the fridge. Okay, so that is kind of a short-term maintenance. Okay, we do it short-term, which means anytime you want to work with the bacteria, you can easily take it out from the fridge and then uh, work with it. Okay. Then long-term preservation, you have two types here. I've stated two types. One is a slant and one is a deep. Usually we just put it in a bottle. Slant. Can you see this?
Sort
Yes.
of.
Okay. So this is a slant. So what is a deep? Same thing. Okay. So what's the advantage of a bottle, of a screw cap bottle with a media here on a slant? Okay. So first advantage is it's a screw cap, so you can tighten it up. You minimize dehydration. Okay. You also minimize the transfer of air. So basically, when there's less air for the bacteria to utilize, they will grow at a slower rate. Okay, so that will allow the bacteria to be preserved longer. So these two the same preparation method. It's just that this bottle is tilted while the agar is still soft, but whereas this bottle is placed straightened up and let the agar harden. So how do you inoculate them? You inoculate them, okay? Whereas for this one, it's just a stab in the middle, okay? So what's the so if you look at this, you will also realize that you can have more nutrients for the bacteria relative to a plate. Okay, a plate will have less media and the bacteria will be a lot growing on top. Okay, however, here the amount of bacteria growing is small. Okay, but the amount of nutrients is a lot, so you can keep them longer. Okay, so that's the advantage of a slant and deep versus a petri dish. So you have so what's the difference between a slant and a deep? What advantages, what pros and cons do you see between a slant and a deep type of preservation? Anyone? Anyone?
Is it like for obligate anaerobes and stuff like that?
No. You see, the main difference is your bacteria here is on the surface, on slant. Here is stabbed inside. So it is easier to pick the bacteria from here. Okay? So you have more surface area to work with versus this deep. But of course, that also means the bacteria here can probably be preserved much longer than a slant. Okay? So those are the uh, main differences. So you these are terms that you should uh, familiarize yourself with so that at least you will be able to understand when you are asked to prepare deeps and slants. Okay. So these are also considered maintenance cultures because you can also use them when you need to work with them. However, they can last much longer, so you don't need to transfer them regularly, maybe about once a month. Okay, so you have more time to do your research and you do not have to spend too much time just maintaining the culture. So usually once you have if you decide to preserve or keep your culture, which means you have decided, oh yes, this culture is important, I'm gonna keep it as a stock culture to preserve it, maybe for work in the future, maybe 1 year or 2 years later. Then you go for long-term preservation. Usually we use glycerol stocks. This is usually a broth, tryptone soy broth, TSB. We add with 20% of glycerol. So what we do is we will culture the bacteria in TSB and then add the glycerol stock and then quick freeze it. Quick freezing is done with liquid nitrogen. So this will bring the temperature immediately to minus 80 degrees Celsius and will be able to preserve the bacteria. Although we are freezing the bacteria here because we added glycerol, glycerol is considered a cryopreservant. So it helps preserve protect the bacterial body even in a frozen state. Okay, so this is how you freeze the glycerol stocks. Okay, next you can have lyophilization. This is a freeze drying method. This one utilizes a freeze dryer, so you have to have a specialized machine. So that's why sometimes if you don't have a freeze dryer, you're stuck with this approach to preserving your bacteria. However, we do have a freeze dryer in our program, so you can freeze dry your bacteria if you feel that they are important. Usually, when we freeze dry a few sets. We do not freeze dry only one one vial of the bacteria. So usually about 5 to 10 vials, because after a few years you will need to resuscitate them and see whether they are still growing. Okay, because you need to continue preserving them. You even with long-term preservation, uh, for glycerol stock, you need to make fresh ones once a year. For freeze drying, fresh ones maybe once every 5 years. Okay, it varies. It depends on how fragile your bacteria is and how important the bacteria is to you. Okay, of if you feel it's very important, then of course you will want to make sure that the bacteria is still alive. So freeze drying is you need a machine. What the freeze dryer do is it extracts the water from your your culture, but at low temperatures, at 4 degrees and below, at freezing temperatures actually. Okay, so how a freeze dryer is better? A glycerol stock, bacteria in glycerol stock is frozen. Okay, whereas a bacteria that is freeze dried does not have any air, it's vacuum packed, doesn't have water. Okay, and when you don't have water, you don't have air, the bacteria is basically in a hibernation mode. Okay, no air, no water, so you can actually just keep it at room temperature. It's a sealed ampule, glass ampule, so you can just keep it at room temperature. So lyophilization is of course the best method available to freeze dry your to preserve your bacteria. Spores are very specific for bacteria that can form spores. If the bacteria can form spores, then it is very useful. You can just allow the bacteria to form spores, and then the spores can. So let's say you have already isolated your bacteria, and you want to work with it. Okay, the first thing you gotta do is you have to identify it and classify it. So identification and classification. What is the difference between identification and classification? So if you have a bacteria, you carry out certain tests, biochemical tests, and you find it to be E. coli. Is it considered identification or do you consider it classification?
Identification.
Yeah, because yeah, identification is for practical purpose. Okay, for example, in the clinical setting or in the food industry. So usually, identification is for bacteria that is considered important in the industry. So in the food industry, Escherichia coli, Vibrio cholera, Parahemolyticus, Salmonella, Shigella, these are important food contaminants. Okay, these are important pathogens that can be isolated from food. So then the process of isolating them and identifying them, so that is identification for practical purpose. However, classification is more than just identification. Okay, classification is for evolutionary relatedness on cataloging. What does that mean? That means, let's just say you have you have let's just say you have E. coli. You isolated E. coli from one place. Okay, but then you want to know how its relatedness to other E. coli and other genera that is near to it from the molecular point of view. Okay, so because an E. coli an E. coli isolated from your study might not be 100% uh, identical to an E. coli isolated from uh, other locations. Okay, so classification is more detailed. You have to compare uh, the molecular sequences for its evolutionary relatedness. So only when you compare many different types of E. coli, then you can see where is a possible source of your E. coli. You know, that's how that's how people that's how classification is done. Okay. For example, a recent study, they looked at the Vibrio parahemolyticus that causes uh, disease in shrimps. So they have the whole genome sequence from Malaysia, they have from Vietnam, China, and even Colombia. So when you compare them, then you are able to say, oh, the one from Malaysia is more related to the one in Vietnam. Okay, so that is how classification is done and how it differs from identification. Okay, so identification is very practical. It's usually few tests are done and easily performed. Okay, so you just need to know, like you got a gram-negative bacteria, you do an indole test, a methyl rate test, Wojcicki Proska, Siemens citrate test. Then from the results, you say, oh, I've identified that it is an E. coli. That's it, done. That's enough, because it is for practical purpose. So identifying it to be E. coli is enough for that, for the for the specific purpose. You don't have to go further into classification unless you are a researcher and you want to know its relatedness and to know where it's from. Okay, so when you do classification, of course you have to do many tests. Okay, exhaustive set of tests. So not only 16S gene that is used, you have some people nowadays, people go for the whole genome sequence of the bacteria, because with the advancement of sequencing technology, you can get the uh, whole genome of a bacteria within 1 week, and the price is probably less than 5,000 ringgit. Okay, so that's how advanced sequencing technology is now. Okay, any questions about this?
No questions.
Okay, so if you have questions, please ask. So, so we know that identification is of a practical purpose. Few tests are done. So the example that I gave you just now, for example, the example I gave you for E. coli, the indole, methyl, Wojcicki Proska, and Siemens citrate test, these are based on biochemical phenotypic properties. Okay, so phenotypic properties. So the problem with phenotypic properties are they can vary. They can vary depending on where you isolate them from. They can vary dependent on at what stage of growth are you testing them. Okay. So, but when you do classification, you rely both phenotypic and genotypic properties. So you rely on more stable properties. So when they are more stable, they are more robust. Okay, so the test done the classification is more robust, because it doesn't depend on one test, and it also depends on tests that are stable. Okay, but when you're doing identification, you're relying on phenotypic properties that are variable, so it is not robust. Your identification, sometimes you have errors that set in. Okay, sometimes you get atypical results. Okay, so that's the difference from between class identification and classification. And also because you rely on less tests, then you have poor discrimination, which means your results is very restricted to either yes, it's an E. coli, or no, it may be a salmonella or shigella. That's all. Whereas for classification, you are not restricted to just a few bacteria, because once you have a lot of once you have both phenotypic, genotypic, and a lot of results from many tests, you are able to basically identify the bacteria from the whole database available. Okay, so the database we're talking about can be your gene bank, can be your RGP. Okay, what I was mentioning is gene bank. So this is the one that they say is NCBI gene bank. The other one is your RDP. So this is now in version 2. So this is a ribosomal database project. Okay, this is a NCBI gene bank. So as both of them are based in America. Okay. So this is where all the researchers will deposit their sequence data. Okay, sequence data in the past it was only like restricted to 16S for microbiologists. Okay, but now it is really expanding all the way to full genome sequences for bacteria and even a full microbiome uh, database. Okay, which means the amount of data here is tremendous. They have microbiome data. Okay, so if you're doing microbiome studies, which means you are extracting the DNA from a specific habitat, then sequencing all the 16S there, and then you upload everything there. So from there you can see the diversity community profile in that environment. Okay, so and of course the amount of data is incredible. It is very big. Okay, so you also have full genome data here uploaded. Okay, so it's really this is a most useful resource if you're for identification and classification. From here also you can download related sequences for your classification studies. Okay, so that's how people can do classification studies. So even if you do only one bacteria isolated from Malaysia, you got the whole genome out, you can then download similar bacteria from other places from the gene bank and then do your um, bioinformatic analysis. Okay, so that is possible now. For RDP, uh, is mainly for bacteria, okay, 16S. So it's considered a cleaner database. So for my lab, we will usually compare our sequence with both this database. These are all freely available. We'll compare with both these two, and then we will conclude the best identification, best similarity for the sequence that we have based on these two. RDP is strong because they clean up the sequence before you can upload it. Okay. Gene bank, sometimes there are errors in the sequence. Okay. But they do curate it and they do clean it up, but this is slower than this. This is phenotypic traits for identification. So these are some of the things that um, you should be doing in your practical. Okay, so first we talk about morphological characteristics. So we talk about the shape of the bacteria, the arrangement of the bacteria, but the cell wall that the bacteria has, endospores and flagella. So these are all familiar to you, right? The terms endospores, right? Cell wall arrangement, shape.
Yeah.
Okay. So these are morphological characteristics of the bacterial cell. Okay, we're not talking about the colony. The colony, as we mentioned in the last week's lecture, it was like we talk about the boundary, the color of the colony, the boundary of the colony, and whether it's raised, whether it's flat. Okay, but now we're talking about the bacterial cell itself. Okay, so these morphological characteristics can be very helpful for identification. Okay, it is not 100% identification, but it's more like presumptive. It helps guide you towards the next step that you need to do in order to identify the bacteria. Okay, so shape. The shape of the bacteria can be cocci, bacilli, spirillum, pleomorphic. Pleomorphic means no specific shape. Varied. Okay, varied can be varied. Okay, no specific shape. So cocci, is supposed to be round. Bacilli, rod shape. Spirillum, like a spirilla. Corkscrew or helical. So cocci, so bacteria that that are cocci will be for example, your Staphylococcus, your Streptococcus. Okay. For gram-negatives, you have your Neisseria. Yeah, mainly Neisseria. Okay. For bacilli, you have your lots of bacteria. Examples: Escherichia coli is a rod shape. Salmonella is also rod shape. For gram-positive, you have bacilli, you have bacillus, you have Clostridium. All these are rod-shaped bacteria. So based on the shape itself, it gives you an idea of what tests that you have to do in order to identify the bacteria. Okay. Then arrangement, whether the arrangement of the cell is in chains or cluster. The best example for this will be your gram-positive Staphylococcus will be the gram-positive cocci that are arranged in a cluster shape. Okay, Staphylococcus is like a bunch of grapes. Okay, so it's cluster shape. Whereas if your cocci is arranged in chains, then it's most likely Streptococcus. Okay, so for cell wall, the gram staining will tell you whether the the bacteria is gram positive or gram negative. Okay. Then for acid fast staining, this one checks whether the bacteria is acid fast. Acid fast staining is based on the presence of mycolic acids in the cell wall. So, do you know any example of acid fast bacteria?
Mycobacterium.
Yes, very good. So, why is Mycobacterium important?
Okay.
So, Mycobacterium is the genus. So, the species you have Mycobacterium tuberculosis and Mycobacterium leprae. Okay, these are the two famous pathogens. So, from the name, do you all know what disease it causes? For example, Mycobacterium. Yes,
Tuberculosis?
yes. Mycobacterium tuberculosis causes tuberculosis. Okay, TB, batuk kering. So, what about leprae? Mycobacterium leprae. Leprae. Any guess?
Leprosy.
Yes, very good. Leprosy custa. Okay, leprosy. So, very, very serious diseases. Okay. So, although at one point of time we consider ourselves to have controlled them, but Mycobacterium tuberculosis is coming back, and yeah, and getting more and more serious. Okay, so we have gram staining, acid fast staining, capsule staining. When you want to look for to see whether the bacteria produces capsule. Okay, we are interested in this because a lot of pathogens actually are able to form capsules, because capsules will protect the bacteria from our immune system. Okay, so most pathogens will have capsule. Okay, wall-less bacteria, bacteria without cell wall will be what? An example? Anyone? So best example would be a mycoplasma. 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 Okay, that is a very well-known bacteria that doesn't have any cell wall. And because it doesn't have any cell wall, the genome size is smaller. Then there are companies in America that are trying to uh, genetically genetically engineered to do helpful stuff with it. Okay, because the genome is small, that's why they are able to. They say it's easier to work with a bacteria when the genome is small. Okay, so you also have atypical cell wall in archaea. So other than shape, arrangement, cell wall, endospores. Okay, the size of the spore relative to cell diameter. What does this mean? So the endospore, some of the endospore formed are smaller than the cell diameter, whereas some of the endospore formed are larger than the cell diameter. So what you see is very strange bulging. So if the endospore is larger than the cell diameter, you see the spore actually bulging out. Okay, and it's it's a very useful characteristic for identification, because among the 2 gram-positive bacteria that produces spore, you have Bacillus and Clostridium. And very strangely, both of them produce spores, but Clostridium spores are always larger than their cell diameter. So when you see spores that are bulging, you immediately know they are not Bacillus, most probably Clostridium. Okay, so it helps in the identification of a bacteria. Okay, so but of course, you know that identification is just not just one test. You have to have a series of tests. Okay, but at least it points you to what tests you must do in order to confirm your guess. Okay, so the next is flagella, whether the location of the flagella, etc. Okay, so these are morphological characteristics that you can use to identify the bacteria. Okay, so whether it's a cluster cocci, whether it's a chain cocci, whether it's a diplococci in capsule, okay, whether it's pleomorphic, this is a pointed shape, pointed ends for fusobacterium. These are curved rods for Vibrio, comma-shaped. These are diplococci, but kidney-shaped, because kidney bean-shaped. So this is most likely Neisseria. These are normal cocobacilli, bacilli-shaped. So can be most of the Escherichia coli, etc. You have spirilla, you have helical. Then the location of your flagella. This is peritrichus, everywhere around the body. This is lophotrichus, a lot at one side, bunch of hair, flagella. This is monotrichus, only one, mono. Then you also have amphitrachus, amphitrachus, one end and another end. Okay, so this is where the location of the spore is also valuable. If it's located central or terminal. You can see that for this one, the endospore is smaller than the diameter of the cell, but for this tree, the endospore is larger than the diameter of the cell. So this is bulging. So typical of Clostridium. Okay. So the location here, this is at the terminal, and this is central. Okay, so they call this a tennis racket shape, and they call this a drumstick shape. Okay, so these are free spores. So you have drumstick and you have a tennis racket shape. So this is the actual stain that you actually see. You can see that the spore is actually bigger than the cell. Okay, so biochemical tests. Okay, so as I have mentioned, we use biochemical tests for the identification of bacteria, and these biochemical tests are usually based on the fermentation of carbohydrates and the breakdown of amino acids. Okay, so in the past we have to prepare all these tests by ourselves, okay, individually, separately, but now we have rapid identification kits that has made the work much more simpler. So here in this course, we will look at enterotubes, three types: enterotube, API, and Biolog. Okay, so what enterotube is? Enterotube is the picture for enterotube. I think is in your practical schedule. So enterotube is shaped like this. Okay, so it has compartments here. So each compartment will have a different test. It's all agar based. So the reaction is locked in an agar. So different types of reaction, different type of tests in each compartment. So here is your inoculation needle. So what you do is you unscrew this. When you unscrew it, you'll find a needle here. Okay, it's a long needle. So what you do is you touch your inoculum here, your bacteria, touch it with the tip of the needle, and then you push it through this. Okay, you push it all the way to the end. So when you're doing it, you're inoculating all the compartments, and then you incubate it and you look for the results. So from this setup, what do you think are the pros and the cons?
Maybe the tons of bacteria will what we call that at the first compartment, but not all bacteria can be found in the last compartment.
Oh, so the one problem that Nuril, is it?
Yes, no real.
Okay. So one problem that he identified is He says the amount of inoculum will be different. Because when you put it in here, more bacteria will be here and less here, right?
Yes.
Okay. Okay. So that is true. So that they have thought about it also. So they have arranged the compartment in a way that the 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 reaction that requires a lot of bacteria, they place it here. Whereas the reaction that requires only, there's maybe more sensitive and will change even with a very low inoculum, they place it here. Okay, so they've rearranged that way in order to circumvent the problem. But still, sometimes if your inoculum is not large enough, you will miss some of these compartments. It's true, yes. Okay, so but this is Enterotube. Okay, so another problem is maybe the agar based. Because it's agar based, it does not last long. It does not keep well. It will dry up. So if when you want to use it, you buy it and make sure that you use it, because unlike others that are dehydrated, this one is agar based and will not keep well. Okay. So that Enterotube has about 15 tests. Okay. Then next one is your API, Analytical Profile Index, from that has about 23 tests. Okay, so the API strips are like this. So these are API strips. Okay, so this is API 20E. 20E, E stands for Enterobacteriaceae. So they are looking for bacteria related to the gastrointestinal tract. Okay, so these are all dehydrated, dried reagents. How you inoculate this is you inoculate your bacteria, uh, resuspending it in sterile saline. Okay, so you're resuspending your bacteria, and then you add your bacteria in. So that will reconstitute your dehydrated reagents. Okay, so you incubate it, and then the reaction will give you a color code. Okay, then the color code, you can compare it with a table, and from there you can identify what kind of bacteria that you have. Okay, so this is a API strip. Okay, so here you can see ONPG, ADH, LDH, a lot of different types of tests. Okay, so 23 tests total. Here you have about 15 tests. Then this is Biolog. This is the most. Yes, 96 tests in a microtiter plate. So it's like a nitro uh, a microtiter plate. So each well is filled with a carbon source, a different carbon source, and then use triphenyl tetrazolium salt as an indicator. So if the bacteria utilizes the carbon source, it will change the color to purple. Okay. So the usefulness of all these rapid identification kits depends on its database. So if you're working with marine bacteria, if you isolated a bacteria from the sea, which do you think is suitable?
Biolog.
Okay, because from here you know that this Enterotube and API is more specific for pathogens, clinically important bacteria, whereas Biolog can be useful for environmental isolates. Okay, but so the strength of a system, identification system that you use is dependent upon the database available. Okay, so even if you have